Hi, now we are going to see about the topic heterochromatin and euchromatin. A chromatin is comprised of 147 base pairs of DNA which are wrapped around a protein called the histone. The organization of the chromatin is not the same throughout the whole genome, but it leads to be condensed in some region or less condensed in some region. The highly condensed area is called the heterochromatin and the less condensed area is called the euchromatin. Here this figure will explain you what is the heterochromatin and what is the euchromatin. The heterochromatin is highly packed area. These are the DNA double helical structure. These are the histone particles. This bead-like structure of the histone particles. The DNAs are wrapping the histone particles. Here it is highly packed, but in this area it is very loosely packed. It looks like a beads on a string like this. This area is called the euchromatin. You can see the heterochromatin. Heterochromatin is the darkly stained as well as the condensed region of the chromatin. It is a tightly packed DNA which is genetically inactive. The heterochromatin does not enable the DNA and the RNA polymerase to access the DNA. Therefore, it prevents the DNA replications and the transcription. The heterochromatin represents less than 10% of the human chromatin. Structure of heterochromatin. The heterochromatin is more tightly condensed form of euchromatin and is also known as 30 nanometer fiber because the diameter of this helically coiled heterochromatin is 30 nanometer, hence it is called 30 nanometer fiber. The formation of heterochromatin includes modification in the histone and the recruitment and spreading of the silencing complexes. These modification causes changes in the structure of the chromatin. These changes will affect the various processes of DNA, including the gene transcription process. The heterochromatin is very stable in structure, but it is also dynamic and changes with the cell cycle. Within the heterochromatin, there are DNA elements called barriers that promote the formation of active chromatin and the exclusion of nucleosomes. This will allow the heterochromatin to spread. The types of heterochromatin. There are two types of heterochromatin. One is the constitutive heterochromatin, another one is the facultative heterochromatin. The constitutive heterochromatins are stable form of heterochromatin because it does not loosen up to form the euchromatin, and it is also contains repeated sequence of DNA called the satellite DNA. It can be found in the telomeres as well as in the centromeres and is usually involved in the structural function. Next one is the facultative heterochromatin. It is the reversible form of heterochromatin, that is, its structure can change depending on the cell cycle and is characterized by another kind of repeated DNA sequences known as line sequence. An example for this facultative heterochromatin is the inactivated X chromosome of the females, that is the bare body of the females. It changes its structure conformation within the cell cycle. Functions of heterochromatin. Heterochromatin has various functions. In the yeast, the heterochromatin core histones are hypoacetylated. This makes the lysine residues more positively charged, which in turn increases the interaction between the histone and the DNA molecules, which making the nucleosome more closed in the structure. And the histone H4K16 is less acetylated in heterochromatin, which promote the folding of the chromatin into high structure orders, further promoting a closed chromatin structure. The heterochromatin is also hypomethylated at H3K4 and K79, which indicates that the heterochromatin is less transcriptionally active. DNA within the heterochromatin is less accessible than the euchromatin, so they are not actively involved in the transcription process. The euchromatin The euchromatin, it is the part of the chromosome which are rich in gene concentration, that is which in DNA, and or a loosely packed form of chromatin is called as euchromatin. The structure of euchromatin is, the euchromatin consists of loosely wrapped chromatin, so the DNA is more accessible for the transcription process. It is also known as bead on a string structure, like this, a bead on a string structure. The modification to the histone tails allow the chromatin to be more open, so they are actively involved in the transcription process. They are active during the transcription. The presence of euchromatin makes the cells transcriptionally active, that is, they are actively transcribing the DNA into mRNA. Structure of heterochromatin. The heterochromatin is more tightly condensed form of euchromatin and is also known as 30 nanometer fiber, because the diameter of this helically coiled heterochromatin is 30 nanometer, hence it is called 30 nanometer fiber. The formation of heterochromatin includes modification in the histone and the recruitment and spreading of the silencing complexes. These modification causes changes in the structure of the chromatin. These changes will affect the various processes of DNA, including functions of a euchromatin. Euchromatin is the part of the chromatin involved in the active transcription of DNA into mRNA. As euchromatin is loosely arranged in order to allow the recruitments of RNA polymerase complexes and gene regulatory proteins, so the transcription can be initiated. The amount of actively producing productivity of the cell is proportional to the amount of euchromatin they are having in the nucleus. We can see the difference between the heterochromatin and the euchromatin. Here in heterochromatin, the DNA is tightly packed, but in euchromatin, it is loosely packed. Here the DNA density is high in heterochromatin, but the DNA density is low in euchromatin. It is heterochromatin is darkly stained, but the euchromatin is stained lightly. Here the heterochromatins are found in the periphery of the nucleus, but the euchromatins are found in the inner body of the nucleus. The heterochromatins show little or no transcriptional activity, but the euchromatins are actively involved in the transcription. The heterochromatins are late replicative, they are compactly coiled, they are sticky, but the euchromatin is early replicative, they are loosely coiled and they are non-sticky. The heterochromatins are genetically inactive, but the euchromatins are genetically active. The phenotype remains unchanged of an organism in the heterochromatin, but the variation may be seen due to the effect of DNA during the genetic process in the euchromatin. The heterochromatin permits the gene expression regulation and also maintain the structural integrity of the cell. The euchromatin result in the genetic variation and permit the genetic transcription. Thank you.